Among the 4 organic compounds in plants, the most diverse and abundant group of organic compounds are the carbohydrates. Plants by photosynthesis produces carbohydrates. Each year, photosynthesis converts more than 100 billion metric tons of carbon dioxide and water into cellulose and other plant products. Animals obtain carbohydrates from plants or eating other animals. However, when the glucose level drops in the body, then proteins are broken down for synthesis of glucose in the body. In human, there is roughly 62% water, 16% fat, 16% protein, around 1% carbohydrates, 1% nucleotide, and 6% minerals. Carbohydrates, similar to nucleic acids and proteins, constitutes of monomeric unit called monosaccharides. When more than one monomeric unit are bond together by glycosidic bond, then they are called oligosaccharides, and more than 20 or so monosaccharides units chain are called polysaccharides. Any compound that comprises more than one hydroxyl group and a carbonyl group are called carbohydrates. If the carbonyl group is at the terminal end of the molecule, then it's called polyhydroxyaldehyde, and if the carbonyl group is present in the middle of the compound, then it's called polyhydroxyketone. So any compound that are polyhydroxyaldehyde or polyhydroxyketone compounds, or a substance that gives these compounds on hydrolysis, are called carbohydrates. The empirical formula for most of the carbohydrates is CH2O. As you can see, the number of carbons in the molecule equals the number of water molecules. The prefixes tri, TETR, and pant, and so on, indicates the number of carbon atoms in the chain, and the suffix OSE indicates that a molecule is a carbohydrate. Each monosaccharide carries a number of hydroxyl groups, to which substituents might be attached or which could be replaced by other functional groups, such as beta-D-glucose-1-phosphate, aldonic acids, and beta-D-glucosamine. The reduced form of glucose, D-glucitol, is also called sorbitol. Monosaccharides with 7 or even more carbons exist in nature, but most are of minor importance. However, one heptose, called pseudoheptulose, plays a major role in fixation of carbon dioxide in photosynthesis, as well as in the pentose phosphate pathway. The different spatial arrangements of atoms is called stereochemistry. Compounds that have same molecular formula and same bonding pattern, but differ in spatial arrangements, are called stereoisomers. In living organisms, the metabolic pathways proceed only with the right rotation of polarized light carbohydrate, rather than the left rotation of polarized light carbohydrates. So, therefore, the understanding of different spatial arrangements of atoms in carbohydrates is very important. Compounds that have same atoms and same bonding pattern, and when placed over one another, gives the same pattern, then these are identical molecules and known as superimposable. But, if compounds have same atoms and same bonding pattern, but when placed over each other, they do not impose the same arrangements, then they are not identical, and are considered non-superimposable. Epimers are carbohydrates, which vary in one position for the placement of the O group. When the molecule has 2 or more stereocenters, then the epimers are diastereomers. So now that we know any compound with same number of atoms and same bonding patterns, but are not identical, being non-superimposable, and can form 2 mirror images, then these compounds are called enantiomers. Any carbon atom that carries 4 different substituents, and exist in enantiomeric forms, are called as the chiral molecules. The stereoisomers, that of chiral centers, is calculated by 2n, where n is the number of chiral centers. So in case of tetrose, which have 2 chiral carbons, that are able to produce 4 stereoisomers. Pentoses will have 8, and hexoses will have 16 types of stereoisomers, and so on. All these stereoisomers have the unique properties, such like ribose and arabinose. Ribose is used for nucleotide, whereas arabinose is used in mycobacterial cell wall as well as substrate, for synthesis of erythroascorbic acid in yeast. As you all know, glucose is broken down in our body for production of energy. Its stereoisomer, D-mannose, is used for preventing urinary tract infections and in treatment of other diseases. The pentoses and hexoses can produce the 2-ring closure structures. In pentoses, C1 binds with C4, giving a structure of closed ring called furanose, and in hexoses, the C1 bonds with C5, giving a pyran ring structure called as pyranose. Isomers that differ in configuration only at the carbonyl carbon are called anomers, such as alpha-D-ribofuranose and beta-D-ribofuranose. To measure optical properties of enantiomers, a single wavelength light, monochromatic light, is passed through a polarizing material, so if the light emerging from the lens rotates clockwise or right, then such compound is said to be dextro-rotatory, designated by a plus sign. And if the light rotates the plane polarized light in a counterclockwise direction, then they're called as levo-rotatory compounds and indicated by minus sign. When a mixture contains equal amount of plus enantiomer and minus enantiomer, then the polarized light is optically inactive, meaning the polarized light rotation will be zero, so, such mixture of equal amounts of enantiomers pair is called a racemic mixture or racemate. Again, these chiral compound mirror image is important, because organisms can metabolize only one of its enantiomers, most probably the D-rotatory carbohydrate. Disaccharides are distinguished, both by the kinds of monomers involved and by the precise glycosidic linkages between them. Sugar compared to fats have low energy, however being water-soluble, it can easily and immediately be used as energy source by the body. So for these properties, the demand of sugar is higher, making you to have more and more sugar. Stored chemical energy is extracted from sugar molecules by the process of oxidation. Energy can also be extracted from sugars, in the absence of oxygen, in a process called fermentation. Bacteria and yeasts carry out fermentation. In plants, glucose is stored in an elongated chain, with two types of polysaccharides, a linear chain of glucose called amylose, and a highly branched chain of glucose called as amylopectin. Starch, the storage of carbohydrates in plants is a mixture of amylose and amylopectin. In animals, fungi and bacteria, glucose is storage in a multi-branched polysaccharide called glycogen. The process of building glycogen is called glycogenesis, and the breaking to glycogen, releasing glycose, is called as glycogenolysis. Like amylopectin, glycogen is a polymer of alpha-1 to 4 linked subunits of glucose, with alpha-1 to 6 linked branches, but glycogen is more extensively branched, on average, every 8 to 12 residues, and more compact than starch. Dextrins are bacterial and yeast polysaccharides made up of alpha-1 to 6 linked poly-D-glucose, all have alpha-1 to 3 branches, and some also have alpha-1 to 2 or alpha-1 to 4 branches. Dental plaque, formed by bacteria growing on the surface of teeth, is rich in dextrins. Cellulose is almost identical to the long molecules of amylose, but with a subtle structural difference. The chemical bond, linking the glucose units of cellulose, differs from amylose only in the configuration of carbon 1 of each glucose. The large amount of glucose energy stored in cellulose can also be used to produce biofuels. Most of the gasoline sold in the US contains 10% ethanol, and some of that ethanol is made from cellulose. Proteins in animal cell membranes have small groups of sugars called attached covalently to them, in such a way that the sugars stick out on the outside of the cell. Proteins with oligosaccharides attached to them are called glycoproteins. They act in cell-cell recognition and binding of other molecules. Cell surface glycoproteins are also important for cross-linking cells and proteins, for example collagen, that adds strength and stability to a tissue. Glycoproteins in plant cells are what allows plants to stand upright against the force of gravity.